Hi student, today you will study patterns of inheritance. In this chapter, we will examine the rules that govern how inherited traits are passed from parents to offspring. You will look at several different patterns of inheritance and investigate how we can predict the ratio of offspring with a particular trait. Also, you will study the behavior of a chromosome during gamete formation and uncover how fertilization accounts for the patterns of inheritance we observe. Genetics. What is genetics? Academic field of studying heredity. Heredity means passing on of physical or mental characteristics genetically from one generation to another generation. George Mendel found that a few simple rules explain many aspects of heredity. George Mendel addressed hereditary factors, which are equivalent of genes nowadays. In mid-19th century, around 1860, while George Mendel was alive, during that time, DNA was not discovered, and there was no concept about gene. Therefore, Mendelian inheritance was based on the behavior of a chromosome. And then the physical trait can be determined by the gene that you receive from parents. In mid-19th century, DNA was not discovered, as I said just before. George Mendel proposed theory of inheritance. He proposed two kinds of law. First law is law of segregation, and second law is law of independent assortment. Okay. So what is chromosome theory of inheritance? In 1866, George Mendel published a paper and stated that parents pass discrete factors onto their offspring. Mendel stressed that the heritable factors, in other words, today we call this gene, retain their individuality generation after generation. Okay? So he is the founder of genetics, and then he was an Austrian monk, priest, and he crossed variety of pea plants and then observed offspring, and he proposed hypothesis that predicted patterns of heredity. And then his proposal could be proved, therefore we accept the theory of inheritance proposed by George Mendel. So he discovered Mendel's law of heredity. So Mendel lived and worked in the Abbey in Austria. He collected large number of offspring from crossing pea plants and statistically analyzed offspring depending on their trait. Mendel's quantitative analysis of the F2 generation plants from thousands of genetic crossed allowed him to deduce the fundamental principles of heredity. At the time, there was no calculator or computer by then. Probably many priests collaborate to make Mendel's theory of inheritance proved. So this person is the George Mendel. He is the George Mendel. And then probably maybe the rest of priests help him to collect the offspring from crossing pea plants, and then also maybe sometimes needed to count the seed, yellow color seed or green color seed, so many number of seed they need to count manually. So that kind of work might be done. Maybe some other priest also help him to establish the theory of inheritance. Then, what principles of inheritance did George Mendel discover by breeding garden pea plant? Mendel dealt with 7 different characteristics of pea plants in his inheritance experiment. First, for example, pea seed shape, the round shape and wrinkled pea seed, or seed color, yellow seed color and green seed color, and flower color of a pea plant, so white flower or violet purple flower, or sometimes pod shape, pod color, or position of a flower and plant size. These are the 7 kinds of discrete characters. Why Mendel chose pea plant? Because it is easy to cultivate. And then the pea plant, one generation is short. Therefore, it does not have to wait for a long time to see the next generation. One of the difficulties to study human genetics is humans' lifespan is long, so it's very hard to collect the data of the next generation. But in case of a pea plant, each generation time is short, so humans can observe several generations offspring from the cross between pea plants. And then also produce large numbers of offspring, so it really helped in terms of statistics. That means his result really provide very little error or mistake. So he used pea plants, and then how he set up the experiment. So Mendel's technique for cross-fertilization of pea plants. Pea plants, this is the flower of pea plants, purple color, or some pea plants show white color flower. Pea plants usually can self-fertilize in nature. So sperm-carrying pollen grains touch on the egg-containing copper of the same flower. So in other words, when you look at this purple flower, for a while, do not look at these seizures. And then if we look at flower inside, you can see this is a stamen. Stamen produce pollen. And this pollen develop into sperm. And this part is ovary. So inside the ovary, there is egg reside inside the ovary. So usually in nature, once stamen mature, then it produces pollen. And once pollen touch, just place on the top of ovary, then this pollen migrate toward the bottom part of ovary. During that time, sperm can produce. This sperm once migrate toward the bottom of ovary, and then there's egg, and then sperm and egg fertilize. And then this fertilization occur within the same flower. We call that self-fertilization. But Mendel want to cross different variant of a pea plant. So first he need to prevent self-fertilization. So what he has done was Mendel chose true breeding plant, that is self-fertilization produce offspring all identical to the parents. So when a purple flowered plant self-fertilize, offspring flowers are all purple flower. When white flowered plant self-fertilize, offspring flowers are all white flower. We call that true breeding or pure breeding. So this purple flower produced by way of true breeding. Also white flower also generated by true breeding. Then Mendel was curious what would happen when he crossed different true breeding plants with each other. So, for example, when this purple flower pea plants cross with white flower pea plant, what kind of a flower color will appear in next generation? Mendel was curious what would happen when he crossed different true breeding plants with each other. In other words, what offspring would result if plants with a purple flower and plants with white flower were cross-fertilized? So to do this experiment, Mendel needed to prevent self-fertilization by cutting off the immature stamen before they produce pollen. So Mendel want to keep only ovary in purple flower, and Mendel only need stamen from white flower. So in purple flower, he cut off stamen. There is no pollen or sperm can be present in purple flower. And inside the ovary, there is oocyte egg. In white flower pea plants, once stamen mature, he use brush and then just collect pollen from stamen and put this pollen placed on the top of ovary. We call that pollination. After pollination, the sperm and egg fertilize, and then this purple flower produce pod, and this pod contains seed. And then Mendel plant these seeds on the ground, and the seeds grew into offspring plants. So the offspring of two different varieties are called hybrid. So again, we call cross-fertilization between white flower and purple flower. And then after pollination, sperm from white flower fertilized to egg in purple flower. And then pod developed, and the inside the pod there is a seed. And then this seed planted on the ground. So offspring of two different varieties, we call that hybrid. And the cross-fertilization, we call this cross-fertilization itself is referred to as a hybridization or simply genetic cross. Before we study Mendel's two kinds of law, we need to study terminology. First, you will hit this sign P, F1, F2. P indicate parent generation. So this P include the true breeding parents. So these P plants resulted from self-fertilization. So true breeding parents are called P generation. P indicate parental. And their hybrid offspring, I said, at the P parental generation, two varieties of P plants crossed. And then the offspring is called hybrid. Okay? The hybrid offspring are called F1 generation. Filial means, in Latin word for filial, it means son, like son or children. When F1 plants self-fertilize, their offspring are F2 generation. F2, second filial generation. It's like a grandchildren generation. Also, you will hit this word phenotype. Phenotype means physical appearance or observable trait. External appearance, physical appearance. So for example, purple flower or white flower, these are the expression of phenotype. Okay? Maybe brown eye or green eye, those are phenotype. Or curly hair or straight hair, those are phenotype. Just to describe external appearance, that is called phenotype. Genotype, gene, the meaning of gene is a hereditary factor determine the difference between purple and white flower of the pea plants. Gene made of DNA. And then what is genotype? Genotype means genetic makeup of phenotype. This genotype determine what kind of phenotype appear. So genotype is the patterns of gene responsible for a particular phenotype in an individual. Genotype is manifested in phenotype. And then genotype can be denoted or labeled by two letters. You can maybe connect this concept as a homologous chromosome. Each trait, certain gene, one gene determines the trait. And then that gene is gonna located on a pair of homologous chromosome. Therefore, you need two letters for the genotype of one trait. So this is a phenotype. Purple color for purple flower, these are phenotype. White flower, these are all phenotypic expression. And genotype means what kind of gene express purple flower. Then two letters present genotype of purple flower phenotype. So when you see two letters like this, large P uppercase letter, and then you see sometimes lowercase letter. What is this meaning, uppercase letter and lowercase letter? I will explain a little bit later, but you have to keep in mind, you have to use two letters for one character. Two letters for one kind of phenotype. The phenotype can be determined by the makeup of a gene, in other words, genotype. So white flower is a trait, and this trait is resulted from white color gene, namely genotype. In this case, genotype labeled as small p, small p. When you look at this purple color flower, the organism's appearance does not always reveal its genetic composition. In other words, it looks like the same color, same appearance phenotypically, but their genetic composition might be different. Look at here, this purple flower carries large P gene, but this purple flower can carry large P, small p gene. White flower has small p gene. Therefore, scientists need to distinguish between an organism's observable trait, like a purple flower or white flower, and its genetic makeup. Therefore, scientists need to know it looks like the same, but their genetic composition might be different. Therefore, scientists put phenotype and genotype. You have to be familiar with to convert the phenotype to genotype. And when you see this genotype, you need to know what this genotype means in terms of a phenotype. So, one example, in giraffe, long necks, large N is a dominant trait. What possible genotype could a long neck giraffe have? So here's what is the character. What is phenotypic character is a long leg. And then long neck is a dominant trait. Maybe there's some giraffe with short neck. But a long neck, that is one character. And then this one phenotype must be expressed by genotype. Genotype must have two letters. So as you see just before, purple flower can have large P. But another purple flower phenotypically looks the same, but their genotype can be large p, small p. But when some genotype has with small p appears in white flower. The question already shows long neck is dominant and it is labeled with large N. Therefore, you have to find at least large N letter should be in genotype. So this and this okay. But if long neck is large N, then small n will be short leg. And this one, at least if you carry one uppercase letter, that also shows dominant phenotype. So this large N, small n, large N will show both long neck. And this one will show short neck. Why? The question already designate large N indicate long neck. Therefore, what possible genotype could a long-necked giraffe has? This is dominant. Dominant always written in uppercase letter. And recessive written in lowercase letter. What is dominant and recessive? You will hear about it a little bit later. But at least in this question, the question already provides information. Long neck is a dominant trait and written in large N. Then what possible genotype. Only you have to think about is 2 letters. And then what kind of 2 letters will describe long neck giraffe. So large N, 2 letter. No problem. Small n, since this is dominant, there might be some counterpart trait. We call that recessive. So you might put some question, right? And then large n, small n, even though it has lowercase letter, it has at least 1 uppercase letter. Then, as you see just before, if you have 1 uppercase letter here, then it shows dominant phenotype. So in this case, the answer can be 2, number A and number C. So terminology. Another vocabulary you have to know about this is allele. Allele means different form of the same gene. The gene for flower color of the pea. And then this is a flower color gene. There is a gene for flower color. But the chromosome, here is chromosome, and then homologous chromosome. So exactly same identical chromosome is here. And then maybe this is the location for the gene for flower color. And then maybe this chromosome at this location, the gene for flower color might carry purple color. And the other element of a homologous chromosome, this location, there's a gene for white flower color. Both are the same location for the gene for flower color. In other words, a homologous chromosome can carry a different version of the same gene. In this case, different version means purple color for flower color gene. In this chromosome, white flower color gene. We call this allele. Allele for purple flower color. We call this region allele for white flower color. In another case is maybe there is another gene here, the same location, there is another gene. And then maybe this is the location for the gene of shape of pea. This chromosome carries allele for round seed. Maybe this region, this location of chromosome carry the gene for wrinkled seed. So this location, we call that locus or loci, is the gene for shape of pea plant. Shape of pea plant seed, actually, plant seed. But one chromosome has round shape, but still this gene express the shape of the seed. And the other element of a homologous chromosome carries gene for wrinkled, is not round. We call this allele. So allele means different version of the same gene, different forms of the same gene. So in this case, what is the same gene? Flower color gene. In this allele is the shape of a pea seed. So variant form of a given gene is called allele. And then this allele has purple color, you can write down uppercase P. And if you decide to put the letter of allele as uppercase B, the other counterpart of allele is going to have lowercase p. You cannot use other kinds of letter. If here, this allele, you decide to put large R for round seed, and the other allele for wrinkled seed should have small r. You cannot use W, okay? Because they are a pair of homologous chromosome, so allele should use same letter, but you can only use this uppercase letter and lowercase letter. And dominant allele, so in this case, large P indicate dominant allele, and then small p indicate recessive allele. Dominant purple flower allele is represented as, here's a large P. And dominant allele always expressed in F1 generation. If two alleles of a homologous pair differ, right now it's different, then one allele determines the organism's appearance, and that is called dominant allele. Only one kind of allele can show up phenotypically. Pea flower has only one kind of color. They cannot show purple and white at the same time. Only one kind of allele can be expressed, especially in F1 generation. We call that dominant allele. Recessive allele, in this case, white flower, this will be recessive allele, and then it will be written in lowercase. The recessive white flower allele is represented as small lowercase p. The trait not expressed in the F1 generation, we call that recessive allele. So white flower color cannot be shown in F1 generation, but it might appear in F2 generation. Same way, here's a round seed. This is a wrinkle. A pair of homologous chromosomes, one chromosome carries allele for round seed, and the other chromosome carries an allele for wrinkled seed. In this case, dominant allele is round. It is going to write down uppercase large R. Therefore, recessive allele, wrinkled, should be small r, not W, because they are still same gene, but different version of the same gene. So you have to share same alphabet. Only difference is uppercase and lowercase. Uppercase represent dominant allele. Lowercase indicate recessive allele. When you want to buy hat, and then there's red hat and blue hat, but they are same kind of hat. Only difference is color. We call that allele. When you want to buy shoes, maybe white shoes and black shoes, those are allele, but they are same shoes. So allele means alternative version of a gene that account for variation in inherited characters. So this is a pair of homologous chromosome. And here's allele for purple flower. Here's allele for white flower. This is the location for flower color gene. So we call this locus, or in case of plural, we call that loci. And then one member of each pair of homologous chromosome comes from organism's female parent, such as mother, and the other member of each pair comes from the male parent. The labeled band on the chromosome represent a gene locus, a specific location of a gene along the chromosome. These are alleles, different versions of the same gene reside at the same locus on homologous chromosome. These alleles may be identical or may be differ. In this case, alleles are differ, but sometimes alleles for purple flower and the other chromosome also can carry purple flower. So alleles may be identical or they can be different. In this case, this is different. These are homologous chromosome, another set of homologous chromosome, another set. So all these four separate homologous pair. If two chromosomes carry identical allele, like large B, it is called homozygous for the gene. If two chromosomes carries identical allele, such as small b, at this location, it is called homozygous. But some homologous chromosome carries different allele, like large b and small b. In this case, also small b, large b. We call this heterozygous. Heterozygous for the gene. In this case, if two alleles of a homologous pair are different, then only one allele determines the organism's appearance, and this is called dominant allele. So this is dominant allele. Therefore, this is recessive allele. In this homologous allele, in this case, only one kind of phenotype appear because they are same. It's the same thing. Small b, only one kind of phenotype appears because there are identical allele. But in this case, maybe large B indicate brown eye, express brown eye, and small b carries maybe green eye. So this is the place for gene for eye color. And then some person can carry this homologous pair. Then that person cannot have left eye shows green color, right eye shows brown color. It does not make sense. Only one kind of phenotype appear, and then between these two different alleles, only one allele can be expressed physically. We call that allele dominant allele. Dominant allele is written by uppercase letter. Therefore, lowercase letter represent recessive allele. Allele for green eye color is suppressed. So in F1 generation, green eye color will not show up. Only brown eye color will appear because brown eye color is the dominant allele. How do you know? Based on this uppercase letter. So let's solve this question. Round seed is dominant to wrinkled seed. If you put the letter large R to denote the genotype of round seed, how round seed and wrinkled seed will be presented genotypically? The question already said large R denote the genotype of round seed. They are asking about genotypical expression. So you have to put two letters. So round C, you have a large R, yes. Counterpart of dominant is recessive, then that is what? Wrinkled seed. Wrinkled seed should have small r. If they have large R, small r, then this large R is dominant. So this dominant suppress small r, so it will show round seed. So one dominant allele is enough to show dominant phenotype. Since there is no large R, it shows recessive phenotype. Therefore, A is not correct. Round seed, at least you carry one large R, that's enough. If we have two, of course, it appears round seed. Wrinkled seed, here, small r, there is no large R. Therefore, there is no allele for round seed. This is the answer. And then round seed, large R, correct. Wrinkled seed, small r, this is also correct. So the answer is C and D. So you need to now convert phenotype to genotype, from genotype to phenotype. You have to know about that. One more practice. Yellow color seed is dominant to green color seed. If you put the letter large Y to denote the genotype of yellow color seed, how yellow color seed will be presented genotypically? So the question looks like how yellow color seed will be presented, but the answer samples shows yellow color and green color together. So the question should be how yellow color seed and green color seed will be presented genotypically. Okay. It's a similar question, a yellow color dominant to green color. And then the question suggests you to use letter large Y to denote yellow color seed. Okay. And then yellow color seed, green color seed is the gene for seed color. And then the gene for seed color has different version. One is yellow, the other version is green. The question suggests to use large Y for yellow. Therefore, green color should have small y. You cannot use G or any other kinds of alphabet. The allele means different version of the same gene. It's still seed color gene. Therefore, large Y is connected to yellow color seed. Therefore, green color seed should have a small y. This green color seed is recessive. Therefore, small y allele. So yellow color seed will show large Y, correct? green color seed, large Y, small y, since large Y is dominant, this yellow color suppress green color. So this genotype will show yellow color. Okay, so this is not correct. Yellow color seed at least carry one large Y, and green color seed, it should have small y, because green color seed is recessive allele. So the answer is D. Genotype means two alleles for each trait. Homozygous, two members of an allele pair in the zygote are the same. Homozygous dominant, homozygous recessive. When you see this same uppercase letter or lowercase letter, that means homozygous. But these two letters both has uppercase, we call that homozygous dominant. If you see two lowercase letters, that is homozygous recessive. Then there's another type of genotype called heterozygous. The members of allele pairs are different. Large R, small r, this is large R is dominant, small r is recessive, and then we call this heterozygous. The phenotypically is the same of large R and large R. Phenotypically same, genotypically different. So what kind of flower color this genotype will show? That is purple. Only the dominant allele achieves expression. Dominant allele means uppercase letter. And the recessive allele is present but unexpressed. So here's recessive allele is present, but it cannot manifest it in physical appearance. So phenotypically, this recessive allele cannot show any appearance. Okay? The phenotype purple flower has two kinds of genotype. What is that? Large R, and large R, small r. If you decide to put dominant allele, I'm going to use large A. Large A indicate purple color, which is dominant. And then recessive allele automatically will have small a. For genotype of homozygous dominant will be large A will label purple color. And small a will indicate white color, which is homozygous recessive. And in that case, heterozygous of a purple color will be large A, small a. So you have a freedom what kind of a letter you will use to express heterozygous. But once you decide to use large A, do not change in the middle of them. Here, homozygous dominant, you use large A, and homozygous recessive, if you want to use small b, then you will mess up. So, question, which of these individuals is a homozygous genotype? Homozygous means two alleles are identical. This is one allele, this is one allele. Homozygous genotype means the alleles are same, identical. And then this is allele on one chromosome, the other allele on the other chromosome in a pair of homologous chromosomes. And then they are not identical. So this is not homozygous. Is this? It's not homozygous. Even this is really wrong way to label genotype. Is this homozygous? They are homozygous, means the same, identical. And then how about this one? It's the same letter like this one, but one is dominant, the other one is recessive allele. We call this heterozygous. Okay? And this also does not make sense. So the answer is this one. Now, Mendel proposed two laws. The first law is monohybrid cross. And the first law is law of segregation. This law of segregation can be explained by monohybrid cross. First, producing pure breeding P generation. So pure breeding P generation means if you cross one purple flower cross with white flower, but this purple flower is self-fertilized. So the offspring is 100% identical to the parents. We call that pure breeding pea plant. Produce pure breeding line in P generation, parental generation. And then cross two pea varieties exhibit alternative trait at P generation. So two kinds of pea plants. For example, maybe purple flower pea plant. The other one is white flower pea plant, and then cross them. And then from a genetic cross at P generation, F1 offspring is generated. Then F1 generation self-fertilized to give rise to F2 generation. So let the plants in the F1 generation self-fertilize. So at P parental generation, and then from cross at parental generation, there's some offspring come out in F1 generation. And then some kind of pea plants produce. Just self-fertilize the pea plants in F1 generation, to get F2 generation. Count the number of each kind of offspring in F2 generation. This is the overall protocol by which Mendel commit genetic experiment. The pea plant produce yellow seed, cross with pea plant, produce green seed, and try to see what kind of seed color appear in next generation. But only deal with one kind of trait. In this case is seed shape. In this case is a seed color. He sometimes cross white flower cross with purple flower to see what kind of a flower produced in next generation. At the time, only he keep an eye on the flower color. So only one kind of a trait focused. We call this mono, one trait hybrid. So for example, this is the phenotypic expression of a monohybrid cross. So at parental generation, this yellow seed, the pea plants produce yellow color seed. And then these pea plants prepared from pure breeding yellow line. The other partner has pea plant which only produce green seed. Also, this partner prepared from pure breeding green line. Once they cross in F1 generation, only one kind of phenotype appears, either yellow or green. Mendel's experiment, F1 generation, all pea seed show yellow color. Once they got progeny from cross at P generation, they got offspring in F1 generation, then self-cross this offspring each other in F1 generation. This means self-fertilization. Observe F2 generation, then you will see most of them dominant yellow seed, but certain small portion also show recessive phenotype. This is phenotypic appearance. We need to convert each phenotype into a genotype. So how you convert the phenotype to a genotype? Indicate genotype of each parent by using letters. Probably in this case monohybrid, so you will use 2 letters. Determine what the gametes are, because cross-fertilization then means meiosis take place. From meiosis, gametes are produced. So the alleles are present on homologous pair of chromosomes. These chromosomes separate during meiosis. Therefore, each allele will be in the gamete. And each allele in the daughter cell is going to fertilize. After reproduction, analyze what the genotype of the progenies are. So first, yellow seed. This is parental generation. The pea plant producing yellow seed cross with pea plant producing green color seed. Okay, so it's a one character. Therefore, you have to use two letters. Yellow seed, as you see here, yellow and green seed cross, and then you will have only yellow seed appear here. That means this yellow color is dominant. And then this is therefore recessive. Since yellow color seed is dominant, you will write down large Y. And then this is a pure breeding line. Therefore, should be homozygous. Once yellow labeled by large Y, then the counterpart of green seed automatically lowercase small y. This plant also produced from pure breeding line, therefore it should be homozygous, small y, small y. Now, for reproduction, to make a next generation, these homologous chromosomes need to separate to form a gamete. So you just half of them, large Y, that allele will goes into gamete. And the other allele also goes into another gamete. But they are same. So you can write down large Y again, but still same. Here is a homologous pair is going to separate during meiosis. One chromosome carrying small y allele goes into gamete. And the other chromosome carrying small y allele also goes into another gamete. So you can think that this is egg or this is sperm. But still same kind of small y, here's large y. So, if you want to write down, you can do that, but it is redundant. The outcome is the same. So this gamete, you think that this is egg, this is sperm, then they fuse. We call this fertilization, and then they form diploid back. So large y, small y, this is the genotype in F1 generation. This F1 progeny supposed to self-fertilize. Self-fertilize means plant with exactly identical genotype is going to cross. Large Y, small y cross with large Y, small y. The possible result from this cross can be visualized with this table. We call this Punnett square. In a Punnett square, the possible gamete of one individual are listed along the horizontal side of the square here, and possible gamete of the other individual are listed along the vertical side here. So from large Y, small y, you just make this separate. So the chromosome carrying large Y dominant allele goes into one gamete. And the other pair of homologous chromosome carrying recessive allele, such as small y, goes into another gamete. It's the same thing. So large Y and small y. Each gamete carries large Y dominant allele, the other gamete carries recessive allele. Then you will write down this gamete here, large Y, small y. These are gamete from female. These are the gamete from male. You write down this one, large Y and small y. The possible gametes of one individual are listed along the horizontal side of the square, and the possible gametes of the other individual are listed along the vertical side. Then you just this gamete and this gamete from female, gamete from male, they fuse together, fertilization, give rise to F2 generation offspring. In this case, large Y, small y. And you just combine them, large Y, small y. Usually dominant allele right before the recessive allele. And then in this case, both small y, small y. Okay. So when you look at the inside of Punnett square, this large Y indicate homozygous dominant. This is heterozygous, still dominant because it has large Y. Large Y, small y also, it is called heterozygous dominant. But this one, small y, is homozygous recessive. So genotype of potential offspring are represented by the cells within the Punnett square. So this is the answer. Okay, let's solve this question. Round seed is dominant to wrinkled seed. If you put the letter large R to denote the genotype of a round seed, what would be the genotype of a round seed plant and what kind of a gamete would be produced from this plant, round seed plant? Okay. This one, this plant means round seed plant. So it already shows round seed is dominant to wrinkled seed. And then this question asks you to use alphabet R, not any other letter. So round seed should carry at least one large R. But genotype always labeled by two letters. Therefore, round seed plant will have large R, correct. And this genotype, small r, no, that is what? Wrinkled seed. Large R, this is correct. And large R, small r, still this is correct because of at least one uppercase that indicate dominant allele. As long as you have one dominant allele, that genotype will show up dominant phenotype, such as round seed. But when you look at from this genotype, you need to know how to pull out a gamete. So in case of gamete, I told you, think about this as a pair of homologous chromosome. So just a homologous chromosome is going to separate during meiosis. So gametes can be large R and large R. The genotype does not have a small r. Then how gamete has a small r? This is wrong. This is already genotypic expression is wrong, so we don't have to see the rest part. The genotype large R is correct, and then gamete is going to be, you can just separate, reduce in half. Okay, so large R or another large R will be in the gamete, but this does not have large R, so this is wrong. In this genotype, since it has uppercase letter, this genotype will show round seed. And then in this genotype, what kind of gamete this genotype produce, we just reduce in half. So one large R and the other one is a small r. So the answer is D. So the first law of Mendel is law of segregation. What is law of segregation? In the F1 generation, all the offspring are dominant and resemble only one parent. Recessive allele were not expressed in the F1 generation, but recessive allele reappear in F2 generation. If I say one more time, recessive allele were not expressed in the F1 generation, but expression of recessive allele come back in F2 generation. Recessive allele is present in F1 generation, but it cannot activate, it cannot produce phenotypical expression, appearance. But still there is a recessive allele there in F1 generation. But in F2 generation, even this recessive allele is expressed and it show physical appearance. About 75%, 3/4 of F2 generation were dominant, and quarter, 25%, were recessive. She said recessive allele expression reappeared in F2 generation. How much? This much. Quarter portion of F2 generation is composed of recessive allele. So the ratio is a 3:1 phenotypic ratio. Genotypic ratio is 1:1. We will see that here. So this is F1 monohybrid. So when F1 hybrid pea plant self or cross-pollinate, which trait appear in F2 generation? We will see that. So this is the procedure of monohybrid by Mendel's experiment. So at parental generation is breeding, pure breeding, purple flower, pure breeding from white flower. And in F1 generation, only one kind of phenotype appear, either purple or white. In this case, purple flower only appear. So in F1 generation, all the offspring are dominant and resemble only one parent, such as purple flower parent. So from this cross, you will have many flowers, many pea plants. All pea plants show purple flower. Now, this purple flower self-fertilization. This purple flower in F1 generation undergo self-fertilization. And then in F2 generation, you can see the rest of this parental expression, such as white flower plants appear. But the ratio is 3:1, 3:1 ratio. So dominant and recessive phenotype appears 3:1 ratio. This is the quantitative analysis. Mendel really plant and then harvest the seed and then plant those seed and then self-fertilize in F1 generation and then get offspring in F2 generation and then count the number of flower, and then he got 705 of purple flowered plants and 224 of white flowered plants. Is approximately 3:1 ratio. So 3:1 ratio is a phenotypic ratio. And he did a monohybrid cross throughout these all 7 characters in pea plants, and then the ratio is always appear 3:1, approximately, or 3:1. A dominant and recessive trait appears a 3:1 ratio in F2 generation. Mendel wondered about what would happen if he crossed plants that differ in both seed shape and seed color. In monohybrid inheritance, he only focused on one character, such as flower color or sometimes seed shape. But he is wondering about what would happen if cross plants that differ in both seed shape and seed color. So in this case, seed shape, round and yellow. And here is wrinkled and green seed. To find out that, Mendel crossed homozygous plants having round yellow seed with plants having wrinkled green seed. Then, in F1 generation, he got round yellow seed. Okay? And then this round yellow seed self-fertilized. And then he analyzed the offspring in F2 generation. So dihybrid means plant varieties having 2 counterparts of characters are crossed. In this case, shape and seed color. Based on this round shape and yellow color, these are dominant allele. Dominant allele always expressed in F1 generation. And recessive allele cannot be expressed in F1 generation. Therefore, green and wrinkled seed are recessive allele. Now, based on this appearance in F1 generation, round and yellow are dominant. So, at the parental generation, this pure breeding round and yellow seed has large R, large R, large Y as a genotype. Okay. True breeding wrinkled and green seed is recessive compared to this round and yellow seed. Therefore, it has small r, small r, small y, small y. Keep in mind, when you decide to use alphabet letter R for the shape of seed, then dominant should be uppercase large R, recessive allele should be small r, small r. You cannot use wrinkle, therefore you will put W. No, be careful. And then yellow seed is also dominant based on that the appearance in F1 generation. Therefore, you have to put large Y here. Then this green seed is automatically small y, small y. Do not put small g, small g. So, you just before this is a phenotypic profile and this is genotypic analysis. Now round yellow seed, large R, large R, large Y, this is the genotype. Large R means seed shape. Large Y indicate seed color, right? And then this seed shape, this phenotype, when this phenotype convert into genotype, you have to use 2 letters. Therefore, round, large R, and seed color, large Y. Since this is dihybrid, you are dealing with 2 kinds of gene, 2 kinds of trait. That's why we call this dihybrid. Now, this is a dominant allele. Therefore, recessive allele is automatically wrinkled seed is small r, and green color seed is small y. They cross, so undergo meiosis, and then they produce gamete. This gamete has allele, 1 allele from this one. In this case, only 1 kind of allele, large R. In the other gene, also, there are 2 alleles, but each allele will be separate during meiosis. So only 1 allele enter the gamete. And then in this case, large Y. And this one also go into another gamete, but we're still same. So only 1 kind of gamete is formed. How about here? What kind of gamete can you get from genotype of small r, small y? Small y plant would produce only gamete with small r, small y. There was only 1 possible outcome for the F1 generation. You just union these 2 gametes, the union of large R, and small r, large Y, and small y. It's better idea to write down same alphabet adjacent each other. If you write down large R, large Y, small r, small y, it's not good idea because we need to figure out gamete a little bit later for F2 generation. So do not do this way. Okay? You have to put same alphabet, same letter side by side. So the union of large R, large Y, and small r, y gamete would yield hybrid heterozygous for both characters. So this is heterozygous for shape of seed and color of seed. I'm sorry. Since it deals with 2 kinds of character, the shape of seed and color of seed, we call this dihybrid. All of these large R, large Y, small y offspring would have a round yellow seed, which has the double dominant phenotype. Therefore, at least this one large uppercase letter is enough to show dominant phenotype. So large R, small r, large Y, small y will appear as round yellow seed. In this offspring in F1 generation self-fertilize with plant which has the same genotype. So Mendel crossed large R, small r, large Y, small y plant with each other, so that Mendel want to figure out whether genes for seed color and seed shape would be transmitted as a package together or not. In other words, Mendel wondered whether the alleles for seed color and seed shape inherited together, or the alleles for seed color and seed shape exhibit independent assortment with the gene inherited independently. In order to get offspring in F2 generation, you have to pull out gamete from F1 generation. Based on this genotype, you have to pull out gamete. And then these gamete, maybe you think that this is maybe female, this is maybe male. So this is oocyte, this is a sperm. What kind of gamete you can pull out based on this genotype, and then this gamete is going to write down this vertical line. And the other, from male, based on this genotype of male, what kind of gamete you can pull out and then write down genotype on this, the top, the horizontal row. How can you pull out this kind of gamete from this genotype? We will look at the genotype first. This indicate seed shape. This genotype indicate seed color. This seed shape has a pair of homologous chromosome, and then each allele, large R and small r, are located in a pair of homologous chromosome. And this homologous chromosome will separate during meiosis and enter in separate gamete. So large R goes into one gamete. Small r is going to go into another gamete. Now, this is a different set of homologous chromosomes. They also will separate. So, large Y allele will enter maybe in this gamete. Then small y allele might enter in different gamete. But there's another possibility. Maybe one gamete has large Y allele, but this time instead of large Y allele, maybe small y allele can enter in this gamete. They are separate set of homologous chromosomes. So they might not necessarily behave together. They have freedom. This gene for seed shape, the gene for seed color, each allele for these two different genes behave independently. So another gamete might have small r from this gene for seed shape. This gamete might include one allele from seed color, this time instead of small y, maybe large Y allele. So the possible result from a cross can be visualized with a Punnett square. I'm not sure how this slide go past this one. So actually, I suggest students write down, pull out the gamete from this genotype, and then pull out the gamete from, if you think this as female, and if this as male, gametes from this genotype, you will write down the horizontal lane, and then pull out the offspring in F2 generation in terms of genotype. So I suggest you do this one. If you have extra paper, please fill in this Punnett square. So it is very important how you pull out the gamete based on genotype. You'll have a similar question on the exam. Okay. And then try not to put gamete large R, small r, large Y, small y. They do not go together. So these are separate genes. Okay, gene for shape, gene for seed color. So only one allele enter the gamete. So some students produce gamete this way: large R, large Y, large R, small y, small r, large Y, small r, small y. It's okay. But if you write down gamete as large R, small r together, this is wrong. If we write down the gamete, large Y, small y, this is wrong. And then this gamete is going to write down in this vertical lane, and then because they are in F1 generation self-fertilized, so it's exactly identical genotype cross each other. Therefore, the other partner also will produce same identical gamete that will write down in this horizontal lane. And then you fuse them, fuse gamete each other, then you will have genotype of offspring. And then that will show this way. This is the answer. You should have these 16 kinds of genotype. The possible result from a cross can be visualized with a Punnett square. Each F2 plant would receive 1 over 4 possible sperm and 1 over 4 possible egg. So for a total 16 possible combination. These are all the genotype of a potential offspring. So based on these genotype, you can tell their phenotype also. So large R, at least it has one uppercase R. This indicate round shape. Large Y indicate yellow color. So round seed and yellow color seed. What about here? Round seed, green color. This is yellow color, but it's not round. It is wrinkled. Based on this genotype, you can analyze the phenotype. So round yellow color seed appears 1, 2, 3, 4, 5, 6, 7, 8, 9. And you will see round green plant appears 1, 2, 3. 3 out of 16 will show round green plant. And 3 out of 16 show wrinkled yellow plant. And then 1 out of 16 show wrinkled green plant. So their ratio in F2 generation shows phenotypically 9 to 3 to 3 to 1 ratio, indicating that the alleles for seed color and seed shape undergo independent assortment. In other words, each pair of alleles segregate independently of the other. So the alleles for seed color and the alleles for seed shape behave, assort, and inherited independently. What does that mean? Look at here. Since round shape and yellow colors belong to dominant phenotype only in F1 generation, it shows yellow color and round shape. One of a parent carries recessive allele. F1 generation, it shows dominant phenotype, but it carries recessive allele in genotype. And hypothesis of independent assortment means whether the color of seed gene influence the behavior of the shape of seed. If the color of seed influence the behavior of shape of seed gene, then they will have only these two kinds of gamete. It's exactly the same in parental generation. Because the parental generation has this gamete, therefore the allele for color gene will always behave with allele for shape gene, but in this time, large Y, large R. Also, same thing, allele for color gene, in this case, recessive allele for color gene, always behave with together with the allele for shape gene. So only these two kinds of gametes can be produced. It's the same thing because this F1 self-fertilized, so gamete from sperm and gamete from egg will be identical. Then in F2 generation, you will have only these 1, 2, 3 kinds of genotype or 2 kinds of phenotype. This is the case of dependent assortment. In other words, the allele for color gene influence the behavior of allele of shape gene. So they behave together like this gamete at P generation. But when Mendel did the experiment, the result is different in F2 generation. How these 16 kinds of combination of genotype can take place, then he analyzed that the sperm from in F1 generation, this genotype, the allele for color gene, allele for shape gene, they independently behave. Therefore, the allele, dominant allele in color gene sometimes enter in same gamete with dominant allele in shape gene. But some gamete has the dominant allele in color gene enter same gamete with recessive allele of shape gene. Also, recessive allele for color gene enter same gamete with dominant allele for shape gene. If yellow color allele always behave with round shape allele, then how this large Y small r or small y large r gamete can be formed? In other words, allele for color gene, allele for shape gene, they do not influence each other. They independently transmit to next generation. Therefore, in F2 generation, you will see this kind of offspring in F2 generation. And then phenotypic ratio is not 3:1. It is 9:3 to 3:1 ratio. From F1 generation, this is F1 generation self-fertilized. And then this, once they cross in F1 generation, they undergo meiosis. When they undergo meiosis, you see this homologous chromosome pair up. The other set of homologous chromosome pair up. The alleles of gene on non-homologous chromosome, so these are homologous chromosome, and these are another set of homologous chromosome, and each other is non-homologous chromosome. Both are these two set of homologous chromosome. We call them non-homologous chromosome. The alleles, such as large R, small r, large y, the alleles of gene on non-homologous chromosome assort independently during gamete formation. So large R and small y enter same gamete, and small r, large Y enter same gamete. Sometimes this large R and large Y can enter same gamete too. Do you remember during meiosis, the independent assortment of homologous chromosome, where this homologous chromosome is going to face to which pole? There is several possibilities to align which pole this each homologous pair is going to face. That is different. We have no way to control. how they are going to face to which pole that is independently associated. Therefore, these four kinds of different types of gametes can be formed. If alleles does not behave independently, you will have only two kinds of gamete. Based on law of independent assortment, in F2 generation, you will have a phenotypic ratio is a 9 to 3 to 3 to 1 ratio. The 9 out of 16 shows dominant phenotype such as yellow round seed. And the second 3 out of 16 ratio shows round and green phenotype. 3 out of 16 also shows wrinkled and yellow phenotype. 1 out of 16 shows wrinkled and green phenotype. So if I ask about in F2 generation from dihybrid cross, you get 1600 seed. How many seed you will get for green round seed? Then this is 3 out of 16. Multiply 1600. Therefore, 300 seed will show round green color. 900 seed will show round yellow color. 100 seed will show wrinkle and green color. Okay. This is the ratio. So depending on how many seed you harvest, and then you multiply this ratio to total number of seed, then you will get the exact number of seed which carrying two kinds of character, two kinds of trait. So the inheritance of one character has no effect on the inheritance of another. Mendel observed that combination of a trait in some offspring differ from either parent. The offspring, what does this mean? But when you see in F2 generation, you will see some same identical of parental generation, the phenotype, this yellow, round, and wrinkle green. You can see identical to the phenotype which you see from parental generation. But this one, these two phenotype, you did not have this phenotype in parental generation. So these are the recombinant type or non-parental type. Okay? Offsprings with non-parental phenotype are called recombinant types or recombinant. 50% frequency of recombination is observed for any two genes on different chromosomes. So new recombinant phenotype or non-parental phenotype are produced due to independent assortment of chromosomes. So the test cross, in other words, can be called back cross. If you see phenotypically, you can say round, yellow seed. But this phenotypic expression does not reveal genotypic composition. So we do not know what is the exact genotype of this round yellow seed. Maybe this one is round yellow seed. But what about this one? This is also round yellow seed too. If you want to know exact genotype, then you have to commit test cross or back cross. So how can you tell the genotype of an individual with the dominant phenotype? So this is dominant phenotype, both dominant phenotype, but you do not know the genotype of an individual. Such an individual could be either homozygous dominant, in this case here, homozygous dominant or heterozygous. Okay? The answer is to carry out test cross or back cross, breeding the mystery individual with the homozygous recessive individual. If any offspring display the recessive phenotype, the mystery parent must be heterozygous. So here, this purple flower can carry large P or large P small p. Either genotype will be manifested in purple flower. But if you want to know what is exact genotype of this purple flower, you will cross this purple flower with homozygous recessive, such as small p. And then if you see only one kind of phenotype appear from that cross, that means this purple flower has homozygous dominant, large P. If from this cross, back cross or test cross, if you see two kinds of phenotype appear 1-to-1 ratio, that means the genotype of this purple flower is heterozygous, large P, small p. That is called back cross or test cross. Okay. Long neck, large N, long leg, large L are dominant trait. Therefore, a short neck will be recessive. Short leg will be recessive trait. Then what possible genotype could a long neck and short leg giraffe have? Long neck will at least have one uppercase letter large N, or maybe both allele can be identical, either one. And short leg, long leg, large L is dominant. Then short leg should not have large L. Therefore, it should have small l, both allele. So do you have large N, small l, small l? Okay. Small n, oh, small n means short neck. So it's not correct. This one is not correct. Large N, correct. Large L is long leg, but the question asking about short leg. So this is wrong. Large N, small l, small l. This is in this case. Therefore, A and D both are correct answer. This is all for the lecture of inheritance.